Today we'll be discussing morphology, reproduction, and life cycle of Volvox. Volvox is a spherical or ovoid multicellular green alga belongs to division Chlorophyta. It is distributed worldwide and comprising about 20 species. Volvox is a cosmopolitan and one of the typical genera of the group. It is found in ponds, andages, and even in shallow puddles. Volvox is one of the planktonic organisms which help to color the water of the pond green. The Volvox colony is of constant size and shape for a given species. Each colony consists of a definite number of the cells arranged in a specific manner and forms an integrated whole called coenobium. So Volvox is haploid in all active phases of its life cycle and has asexual as well as sexual reproduction. Next is morphology. Volvox cells are spherical or ovoid. You can see here in this diagram, having a cup-shaped chloroplast, two flagella, nucleus, vacuoles, and an eye spot. Eye spot helps with light perception and contributes in phototaxis. Volvox coenobium. You can see here this diagram. Volvox coenobium is the largest and most highly differentiated of all the free swimming colonial forms. It looks like a minute green ball, the size of just a small pinhead, varying from 0.5 to 2 mm in diameter, and therefore can easily be seen by naked eye. It is a sphere of extracellular mucilage or matrix with a firm bounding layer. It is a sphere of mucilage or matrix with a firm bounding layer. It contains many small biflagellate somatic or vegetative cells. You can see here these small are the vegetative cells. In addition, there are few large non-motile reproductive cells called gonidia, mostly in the posterior pole. The reproductive cells never develop functional flagella and never contribute to any somatic function of the individual. They are entirely specialized for reproduction. In some species, the reproductive cells are derived from cells that originally look and function like somatic cells before they enlarge and divide to form new progeny. Depending upon the species, the number of the cells in the colony varies between 500 to 50,000 cells. Each cell has its own gelatinous sheath or mucilaginous envelope and thus separated from its neighbors by a considerable expense of gelatinous material. And this shows that Volvox is not an individual but an association of number of independent cells. In majority of the species, each cell is connected to its neighbors by a series of broad protoplasmic strands. As you can see here, these are protoplasmic connections. Next is reproduction. So Volvox is haploid in all active phases of its life cycle, but it has two types of reproduction, asexual and sexual reproduction. All the cells of the young colony are green and vegetative in function. These are somatic cells. Then, in each adult colony, a certain number of the cells, especially in the posterior part of the colony, enlarge and enlarge considerably and function either as asexual or sexual reproductive cells. So these cells are recognizable by their large size. You can see here, large size, definite large nuclei, and more dense granular protoplasmic contents. Adult Volvox colony exhibit a degree of polarity and differentiation of cells accompanied by reason of labor. So it is indicated by the position of the vegetative and reproductive cells. The anterior vegetative cells have large eye spot. This is an eye spot that guide the colony to light while the reproductive cells posteriorly can reproduce. So as the coenobium has all its reproductive cells either entirely asexual when the season is favorable for growth, or sexually toward the end of the growing season. So first of all, we'll discuss asexual reproduction. The daughter colony, or gonidium, grows from the reproductive cells around the equator of the colony. So each special reproductive cell gradually pushes back into the colony, withdraws its flagella, increase in size, and becomes more or less round in form, right? So this is gonidium. So each then undergoes about 11 successive longitudinal binary fissions, forming a daughter coenobium within the parent cell wall. The first 5 divisions are asymmetrical, resulting in an embryo consisting of 32 cells of similar sizes. The first division of the cell is longitudinal and in the anterior posterior plane in respect to the parent synovium as whole, and the second is at right angle to the first. After that, each of the daughter cells divide again and forming a curved plate. I can see here. This is E diagram. This is 8-celled, curved 8-cell plate. Now, then the 16-celled stage is arranged, this F diagram. 16-cell stage is arranged in a hemisphere called plachia. So this is plachia, in which the flagellated ends of the cells are directed toward the interior of the plachia. Now, fifth cleavage produces 32 cells, and the sixth division is asymmetrical since the 16 most anterior cells, one half of the 32 cell means, typically cleave unequally to produce 16 cell pairs of unequal sizes, 16 large gonadial means reproductive cells and 16 smaller somatic cells. The larger cells will become the new reproductive cells. Finally, at the end of the 11 cell division, the embryo is a hollow sphere with one layer of the cells whose flagella are directed inward, and the sphere has a small aperture. This G diagram has aperture called phyalophore, which is directed to the exterior of the mother synovium. Now, at the end of the division stage, all the cells are in contact with each other. So, for the colony to be motile, flagella must be directed to the exterior of the plachia. So, a fascinating process corrects this. Just look at this H diagram. Corrects this orientation in the life cycle of Volvox. This is called inversion, right? So, in this process, the young daughter means colony turns inside out through the phyalophore means aperture to bring the flagella from its interior to its outer surface. And the mechanism of this process is a series of cell shape that changes and causing the multicellular sheet to bend outward, and the bend propagate from the interior to the posterior pole, eventually resulting in an inside-out spherical sheet of the cells. And inversion together take about 8 hours, and the complete asexual cycle takes precisely 2 days when it is synchronized by a suitable light-dark cycle. About a day after it is done, the well-developed daughter colonies bulge into the hollow of the parent colony, where they remain for some time before they are set free. Both the adult colony and the daughters within means increase in size, but without further cell division, by depositing large quantities of the matrix. So these daughter colonies usually escape by rupturing of the parent wall, and they are enzymatically released from the parent and swim away. Next is sexual reproduction. Sexual reproduction in the flagellates has rarely been observed, other than in volvocales. The volvocales display all gradation of sexual reproduction from isogamy to highly developed heterogamy. So in Volvox, it is of advanced type, whereby true eggs and the sperm develop from the special reproductive cells. Therefore, the reproduction is of oogamous type. So, Volvox carteri reproduce asexually most of the times, and in nature, it reproduces sexually once each year. As in asexual reproduction, reproductive cells around the equator are specialized to develop into germ cells. So most species have male and female colonies, uh, two types of colonies. However, in some species, the colonies are hermaphrodite and are this able to form sperm as well as ova, although not at the same time. The reproductive cells that will develop into male gamete called antheridia, and the antheridia undergoes repeating fission to produce rounded, oval-shaped sperm packets, which are sort of loose aggregates. So it is astonishing that such a lower organism has a device to preserve and protect its sperm for a longer time, as in the case of spermatophores of metazoa. So the sperm packets is a flat packet of 16, 64 or 128 constably modified sperm, each in cylinder, pale, and bears a pair of flagella in the middle of its length. Upon the liberation of sperm packets from the parental colonies, they swim away as a unit to a receptive colony in search of eggs. So, on the other hand, egg dwell from the specialized reproductive cells that have not undergone division. A few macrogametes means eggs are large and loaded with food for nourishment of the young colony. When these macrogametes are mature, means egg matures, the sperm packets enzymatically lyses a hole in the colony and dissociate into individual sperm, which swim slowly through the gelatinous oogonial wall. The sperm enters the cell properly through the beak-like receptive spot and fuses with the egg nucleus to form zygote. Then the fertilization means this type of fertilization is internal, where the ovum is fertilized without becoming detached from the parent synovium. So eggs that have not been fertilized might dwell parthenogenetically, thus forming daughter colonies. So soon after fertilization, zygotes secrete a thick three-layered, smooth, spiny protective outer wall and becoming an oospore. So this function as a resting stage. Meiosis then occurs during this resting stage, and the zygote produces four spores. Then the parent colony decays and the oospores fall into the bottom of the pool where they survive where they survive the winter. So upon the restoration of favorable circumstances, oospore germinate and each meiospore either develop to form a new colony or develop as a biflagellate cells that later divides into a small colony. So just look at this diagram. Here, this has been sexual reproduction. Here you can see here this A. This is antheridium. And from B to C diagram, they are showing stages. Means these stages showing development of sperms. So here are the sperms. Then look at this second diagram. Here, this is oogonium A. Oogonium and means with immature egg. Then B, this is with mature egg. C, this is showing fertilization. And D, containing an oospore. Thank you.